Hello, I'm David Kerr, Professor of Cancer Medicine from the University of Oxford. I mean, all of us have been enthralled with the Nobel Prize winning science that underpins this extraordinarily renewed interest in using the body's immune system to recognize and destroy cancer cells, all precipitated by the fantastic work of immune checkpoint inhibitors and the various pathways involved in this, with an incredibly burgeoning new, reinvigorated, let's call it, area of science. I thought it was worthwhile us together perhaps revisiting the cycle of cancer immunity, so-called, and thinking where along that cycle we might be able to intervene. Because I think the future is that we're going to see increasingly complex, but I hope logical immunotherapy combinations to treat cancer. And the more we understand of the processes, the pathway involved in immunological recognition and destruction of cancer, the more focused, the more rational our therapeutic interventions might be. So in terms of the immunity cycle, the cycle starts with presentation of cancer-associated antigens to the body's antigen-presenting cells, and that's mediated by the major histocompatibility complex. These molecules that sit in the cell surface with bits of the antigen nestled within them. Where do cancer-associated antigens come from? Classically, genomic alterations, mutations, translocations, read-through frame shifts, and so on. They can come from cancer-associated proteins which are normally expressed at immune-privileged sites. It could come from viral protein, step one. Step two, the antigen-presenting cells, upon capturing the cancer antigen, then migrate to secondary lymphoid organs. There, the third step, these antigen-presenting cells, particularly important to the whole process, they prime and activate naive T cells. These are the sort of fighter pilots of the immune system, if you like. Well, when I was my first day in medical school, we were spoken then to the dean, who said, some of you will become physicians, some of you, the brave few, will become surgeons. He was a surgeon. He said, physicians are the bomber pilots of medicine, but we surgeons, we are the fighter pilots. And that's always stuck with me. So the APCs prime activate naive T cells, and again, it's through this MHC antigen T-cell receptor interaction that switches and activates the T-cells, along with a whole host of other co-immunostimulatory molecules on both cell surfaces. The activated immune cells then enter the tumor, then enter the circulation, step 4, and through an interesting process, rolling along the endothelial cells into the tumor microenvironment. They there recognize the tumor cells through the interaction of the T-cell receptor and its cognate antigen found in the surface of the tumor cell. So this is a cycle coming back again. And there, when appropriately activated in the correct immune microenvironment, kill the target cancer cells. So there's a cycle of immunity, of recognition, of transfer of data from one cell to the other, and of those killer T cells in the right microenvironment, being able to recognize and destroy the tumor cells which express the appropriate antigen to very specific interaction. And what we can see is along that different pathway where we can intervene therapeutically. Can we create neoantigens? Can we prime new neoantigens? There's some evidence to suggest that this may indeed well be possible. Priming neoantigens from the dark genome, so-called long non-coding RNA. Can we increase presentation of these neoantigens by the by MHC system? Yes is the answer. We have some very nice evidence ourselves suggesting that whether there are some drugs which can hugely increase overexpression of MHC, hugely increasing the potential for any neoantigens in the cancer cell to become available to the to antigen-presenting cells. And clearly there are possibilities around vaccination and priming and boosting all along the system. If we can identify these unique neoantigens, we can use vaccines subsequently to boost the APC antigen response and therefore boost that whole cycle of immunity. Clearly understanding about the immune checkpoint inhibitors and that interaction between the antigen-presenting cell and T cell, and blocking some of the factors that might reduce the interaction between the T cell and antigen-presenting cell, the immune checkpoint inhibitors being an absolutely fantastic example of that. We understand less about the holding trafficking signals that allow that migration of activated T cells from secondary lymphoid organs through the circulation into the tumor microenvironment. But we're understanding a lot more about the tumor microenvironment and how we can make that more permissive for the T cells to be effective in terms of killing any of the tumor cells. We're understanding more about the cytokines involved, the chemokines, the interaction between different subclasses of T cells, so that we can create or identify an immune permissive microenvironment. And if we start to put all that information together with different elements of therapeutic intervention around the cycle, that's how we start to create quite rational combination immunotherapies, which could be truly synergistic in the old pharmacological sense. And one of the beauties of these approaches is the fact that the side effect profile of the immunomodulating agents is usually not overlapping, often mild if we can get the dose right, and therefore it does offer a real potential for pretty safe combinations in terms of pushing it further forward together. So I hope this wee talk gives you a sense of the complexity of the system, but also of an increased understanding of it, always seeking the Achilles heel, always seeking the therapeutic lever that allows us to integrate and put together some of these remarkable insights that we're gaining to the immunity cancer cycle. Listen, thanks for listening as always. I would be very interested in your own comments and own ideas as to where we might intervene to block that cycle, or at least to augment the positive elements of it. Thanks for listening. For the time being, the escapers, over and out. Thank you.